Welcome back to this channel. This morning we started the basis of classification of the animal kingdom, and this evening we are going to be talking about the other basis of classification. We've only discussed the habitat as a basis for classification. We've discussed cell organization, symmetry, body plan. All right. This evening we're going to be talking about the germ layers as a basis of classification, the segmentation. Okay, let me list what we're going to be discussing um, as the basis of classification. We're going to talk about the germ layers as a basis of classification. We'll talk about segmentation as a basis of classification. We'll talk about possession of coelom. We'll talk about skeleton. We'll talk about notochord. We'll also talk about the kind of cleavage. The kind of cleavage. We'll talk about the type of development following the cleavage, the type of development. Then we'll talk about the types of egg, the type of egg. Then we'll talk about the body temperature, body temperature. We'll talk about the mode of digestion and digestive system. The mode of digestion and digestive system, mode of respiration and respiratory system, circulatory system, excretion and circulatory system. Okay, so we are going to use all of this as our basis for classification. We're going to explain how we use them to classify organisms. All right, let's begin with the germ layers. All right, so what is germ layer? Germ layer basically the word germ actually refers to the to cells that are pluripotent, that can develop into something else. That's the meaning of germ layer. That can develop into the organism or develop into a body part. That's the meaning of germ layer. Is that clear? Okay. I did not want to introduce a new word. That's why I didn't use it. So we have germ layers. Based on germ layer, organisms are classified into two basically, and they are the diploblastic and the triploblastic. Okay. Now the diploblastic, from the word di, it means two. It means that they have two germ layers, which are the endoderm and the ectoderm. Okay? So, and the endoderm and ectoderm are separated by a mesoglia. Now, mesoglia is not a germ layer. Why? Because these two layers are layers of cells. Is that okay? They are composed of cells. The endoderm and the ectoderm are composed of cells, while mesoglia is the non-cellular layer. In fact, it's composed of just fluid. So it is the fluid portion that separates between the endoderm and the ectoderm. So organisms that are diploblastic have only these two germ layers, and an example is the netophores, the sponges, netophores, coelentrates, and netophores. Okay, the jellyfish, the sea anemones, anemones rather, they are here, exactly. So they are diploblastic. Then the second type of germ layer that we have is the triploblastic organism. Triploblastic organism, from the word tri. Tri means three, right? So it means that they have three germ layers, which are the endoderm, the inner layer, the endoderm, the middle layer, which is the mesoderm, and the outer layer, which is the ectoderm. Okay? Now, in the other video where we talked about the general characteristics, we already talked about what these germ layers become. So the endoderm is the one that becomes the digestive, the GIT. It forms the GIT. It forms the respiratory system, you know, while the mesoderm is the one that forms the glands. No, the mesoderm forms the liver, yes, the liver, the organs. You understand? It forms the organs, the liver, the kidney, you know, that is the mesoderm. The kidney, the, which other thing is there? The intestine is endoderm. So then the ectoderm is the skin, the nervous system. So mesoderm forms liver, kidney, reproductive system, while the endoderm forms the GIT, the respiratory system. Yeah. So that's it. All right. So that is about triploblastic organisms. I've discussed this in the other video, so I'm going to link in the description. So endoderm, mesoderm, and ectoderm, triploblastic organisms. And beginning from using our mnemonic that we already learned, P, C, C, P, N, A, N, NAMA, E, H, C. Okay? From the P, the second P, which represents the plathiomints, to caudate, they are all triploblastic. I repeat, from these guys, the plathiomints to caudate, they are all triploblastic. Okay, I already made another video on the classifications too. So there's a way we use these characteristics to broadly classify them into a tree-like classification. So I'm going to link that video in the description too. So that's based on germ layer. So the next one is segmentation. So let's classify on basis of segmentation. Now, organisms are usually, the animals rather, animals are segmented. They have body segmentation. And what is segmentation? Segmentation simply means separation or divisions in the body part. Is that clear? There are divisions in the body part. And if the organism is fully segmented, or so if the organism is truly segmented, then we say the organism is metamerically segmented. Now, what is true segmentation? Okay, true segmentation, metamerically. True segmentation is also called metamerism. Exactly, that's what I'm supposed to write. Metamerism. Okay, so metamerism is when an organism is segmented both internally and externally. So what that tells us is that an organism could either be truly segmented, that is metamerically segmented, or only internally segmented, or only externally segmented, or pseudo segmentation. So we'll call it pseudo-metamerism, okay? Pseudo-metamerism, that's the word. Pseudo-metamerism. Now, what is true segmentation? That is true metamerism is the one seen in organisms like annelids. Why is that true segmentation? It is true segmentation because it is both internally and externally. Remember I said segmentation simply means what? There is what? Repeated portion. The body is separated into different repeated portion. More like there are repeated portion of body parts, exactly, such that each body part has the same repeating units. Yes, that's what we'll say, repeating units. And those repeating units give rise those repeating units give rise to an elongated body, exactly, elongated body parts. All right, so if it is happening internally and externally, as we see in annelids, primarily earthworms, that is true metamerism or true segmentation. Then, if it happens only internally, as we see in chordates, then that is internal segmentation. Then there is external segmentation that we see in insects, arthropods. Then there is pseudo-metamerism that we see in tapeworms. Tapeworms are pseudo-segmented. What? Or false segmentation. Now, what false segmentation means is this, that the embryonic stage of the organism is not segmented in its embryonic stage. The segmentation only happens in the adult. Is that clear? So because of that, we say it is not true segmentation, it is false segmentation or pseudo-metamerism. And that is it for segmentation. So let's see coelom. Okay? Now, coelom refers to the body cavity, exactly. So, an organism that has coelom is said to be coelomate. Okay, based on body cavity, we're talking about classification. We classify organisms into three. We have the acoelomate organisms, the pseudocoelomates, and we have the eucoelomate. Coelom, that's eucoelomate or coelomates. The word "eu" actually means true, exactly. So we can say eucoelomates as well. So acoelomates are organisms that are not coelomates, not coelomates. Why are they not coelomates? Because they have no body cavity. They don't have what? Body cavity. Then the pseudocoelomates are the ones that have true body cavity, and these ones, the coelomates, these ones have false body cavity. Sorry, pseudocoelomates have false. Pseudo means false. Coelomates, that is, they are organisms that have body cavity, but in this case, it is false body cavity. I'm going to explain what that means. Then eucoelomates are the ones that have true body cavity. Okay, so embryonically, during the embryonic development of an organism, you already know during embryonic development, we have morulation, we have morula, blastula, and gastrula, right? Okay, so as the body is undergoing division, it is forming it is forming into these embryonic stages. Is that clear? So now, during these embryonic stages are basically aggregates of cells, isn't it? So those aggregate of cells, they begin to form into shape. First, they form into a spherical shape, exactly, those aggregate of cells, and then they begin to form what is called an enteron. Exactly. So an archenteron actually. So that is the base of classifying into, when we talked about body plan the other time, we mentioned protostomes and deuterostomes. So I'm going to be using deuterostomes, which we are as humans. So the second opening, the archenteron is the first opening. The second opening is the one that forms the mouth, right? And the first opening forms the anus for deuterostomes, okay? Now, as they now, as the cell begin to organize themselves in that way and arc inward, form inward, that cell, that inward formation there is going to result in a series of hollow, hollow, hollow aggregate of hollow set of cells, hollow cells, set of cells that will form the endoderm. Then we are going to now see the outer layer of cells. These are series of cells, is that clear? Which will form the ectoderm. So this will be the endoderm. Now, you see, if this were a diploblastic organism, this place would be the mesoglia. But there is a mesoderm, because this guy is going to be, the deuterostome is going to be a deep triploblast, okay? So there is a mesoderm, and in this case, the mesoderm is going to be lined by a cavity. The mesoderm is going to be lined by a cavity, and that cavity will exist between the mesoderm and the endoderm. So there's going to be this cavity or space, cavity or space. So that's what we call the coelom. Is this clear? So this is lining the space, this is the space between the endoderm and the mesoderm, okay? So you can see that there's another aggregate of cells here, group of cells, exactly, here. And those group of cells, I've told you, form the body parts, isn't it? Okay. So now this guy, this cavity here is the one that gives rise, that makes space or makes it possible for the development of other internal organs, creates space for other internal organs to develop within, exactly. And of course, this cavity also makes it so that makes it possible for larger size in organisms, especially chordates. That's why chordates have large body size, okay, because of the coelom, all right? While the pseudocoelom is when the mesoderm is not the mesoderm appear only in form of what we call mesodermal pouches. Mesodermal pouches. So it is not lining the entire wall. We call it the peritoneum. We call it the peritoneum. The peritoneum. Peritoneum. That is lining the gut. You know, the gut is formed from the endoderm. Is that clear? So because this guy is not, the mesoderm is not lining the body cavity, the coelom rather, is not lining the peritoneum in this case. In the case of pseudocoelom, it is not lining it. It is only forming as pouches. So we have the outer guy, the inner guy here. Then you now have mesodermal pouches all around. So this is pseudocoelom, mesodermal pouches. So these are our endoderm and our ectoderm. Is that clear? So this, unlike here, where we have the coelom covering the entire having a whole section here to itself, so that this will be the mesoderm throughout here, mesoderm, ectoderm, then there is the endoderm. So in this case, this is true xylem, this is pseudoxylem, this is mesodermal pouches. Does that make sense? Then the acoelomate is where there is no body cavity at all. And that's why they have reduced body size, the organisms that possess this kind of, that are characterized as acoelomates. Okay? So, and an acoelomate is platyhelminths. The flatworms or platyhelminths are what? Acoeloms. Acoelomates rather. So an organism can be triploblastic yet be acoelomate. Coelom is body cavity. So the presence of body cavity is what makes an organism coelomate. So, but platyhelminths are triploblasts, but they are not coelomates because they only consist of a mass of cells, thick mass of cells from the inside to the outside. There is no space. Unlike us humans, for example, that have spaces in between, inside for our organs, the liver, the kidney, the stomach, the spleen, and all of that to stay. Exactly. So those are spaces in between our gut or our respiratory tract, which form the endoderm, and the body wall, our skin, our muscles first, our muscles which form the mesoderm, and our skin, the outer guy, which forms the ectoderm. I hope that is clear. Then let's go to the skeleton. I wanted to explain something on this. So maybe when we get to cleavage, I'm going to explain them again. So we said, okay, so I didn't mention the nematodes are coelomates. This one, acoelomates. Then from annelids downward, coelomates. Is that clear? Acoelomates, annelids downward. I've mentioned this also in that other video, but that one is for broad classification. So it's going to help you. There's no way you go through all these videos and not remember them, because I keep showing you tricks and how to remember them based on the way we classify. All right, so we have skeleton. Based on skeleton. Now, skeleton is basically the body framework that helps to support and give rigidity to the body. Is that clear? So our skin, our flesh, our muscles are just flaps that are hanging or covering a particular skeleton. So it's the body framework for support and structure that is the skeleton. Okay. Now, based on their appearance, where they appear or how they appear on the body of the organism, not form now, not the content, but actually how they appear on the body of the organism. We have the endoskeleton, then we have the exoskeleton. Now, what is endoskeleton? Endoskeleton is when the organism has its skeleton, its bones or rigid form structure inside the body, such as we have in humans, we have in mammals, most mammals, okay? Endoskeleton. The body framework is internal, while exoskeleton is the body framework is external, such as we have in insects primarily. Insects have exoskeleton. Many arthropods, these guys, arachnids, what do you call them? These guys, yeah, arachnids. Under the arthropods, there's these particular guys that most arthropods, if not all arthropods, actually have exoskeletons. Okay, I was trying to remember prawns. This family, this group class of prawns. Crustacean hair. Crustaceans. I was remembering this crust actually means hard shell, hard exoskeleton. Exactly, hard covering crust. Do you understand now? Crustaceans. Okay, so those are the exoskeletons. Now there are other organisms, and those are under these guys. Now there are other organisms that actually possess what looks like exoskeletons too. For example, in the poriferans, you see spicules. Spicules act as spicules. They act as exoskeleton metaphors. Have the entire structure is rigid because it is made of calcium carbonate. Exactly. These guys, they form corals, so we call them corals. So they are made up of CaCO3 exoskeleton. You understand? These guys, mollusks or some mollusks actually have calcareous. Yes, this is also calcareous. Is that clear? Calcareous or calcareous. That's the pronunciation. Calcareous. The word calcareous means having to do with CaCO3. So mollusks also have exoskeleton in form of shells. Is that clear? Not all mollusks. Some have their shells reduced, some have it internally. Okay. So, but snails, as we know them, snails have calcareous exoskeleton. And of course, humans also have some they are forms of skeleton, such as our nails, exactly. They are modifications, yes, of skeleton. We have some mammals like goats, ram, those guys, bulls. They have horns. They're also extensions of skeleton that are exhibited outside, okay? So that is based on where they appear. Then based on their forms, actually their content, what they are made of. We have bony skeletons, we have cartilaginous skeleton, and hydrostatic and chitin. So bony as exoskeleton that is made of bone, cartilaginous made of cartilage, hydrostatic, this one made of chitin, then hydrostatic is just made of fluid, okay? All right, so those are the classifications. Then the next one is notochord. So classification based on notochord. First, what is notochord? Notochord is basically an elongated structure that is found dorsally on the body of chordates, and that's why we call them notochord, chordates, exactly. So it is not found on all of these guys. These guys don't have it. No, from here downwards, they don't have it. They don't have it. Okay, so this one is called hemicodata. Exactly. In fact, they are not chordates. Hemicodata is not chordates. Chordates have been divided into two: protochordates and vertebrata. I'm going to give you the. Yeah, I think I will link the video in the description. I mentioned that already. So notochord. Notochord is that elongated and is not divided, is undifferentiated. You understand? Structure that you see on the dorsal portion, dorsal to the body of the animal, exactly. So that's the notochord. All right. So, okay. So there are animals that are chordates and there are animals that are non-chordate. So these guys from PCCP to hemichordates, they are non-chordate, while chordates are um, they possess notochord. Okay, so that's that. Then the next one is the cleavage, the type of cleavage that organisms undergo. So we have cleavage, okay? Cleavage simply means the continuous mitotic division that takes place immediately after there is fertilization. You know, when fertilization occurs, then the zygote is formed. Yeah, the zygote is formed. So the zygote is a single cell, right? So the single cell begins to undergo mitotic division several times. Okay, and it will give rise to the morula, with double L or one L. Then this guy gives rise to the blastula, and this guy gives rise to the gastrula. Then from here, different of the body parts begin to emerge, right? So my point is now cleavage, okay? Now cleavage is how at the embryonic stage, the organism arise from the mass of, how the embryo develops rather from the mass of cells, this mass of cells that are formed, how the organism develops. All right, so there are two types. We have the mosaic or determinate cleavage. Determinate cleavage. While there is the non-mosaic or the non-determinate or indeterminate, that's what it is, indeterminate cleavage, okay? So cleavage simply means multiple splitting, right? I've been following this channel where we talked about in chemistry, cleavage, splitting, all right? So now in the mosaic splitting, during the embryonic stage, the blasto, now those cells, those cells that are formed in, for example, if this is a blastula, so these cells are called blastomeres. Now this spherical structure is what we now call the blastula, is that clear? So this structure here, this mass of cells, once these cells do not separate, for mosaic cleavage rather, for mosaic or determinate cleavage, once these cells do not separate, then an embryo can emerge from here. Now this is the reason. Each of these cells have a determined role. That's why it's called determinate. They have a predetermined role. So if they separate, it means that they can no longer play the role that they ought to play once the embryo is formed. So the embryo, none of the blastomere, those are the small cells, none of them get separated. Is that clear? So that is the mosaic cleavage. Then the non-mosaic or indeterminate cleavage is the one that occurs with us. So this one occurs in annelids, the mosaic cleavage. The non-mosaic cleavage occurs in mollusks and chordates, especially. So now the mass, our blastula, this spherical mass of cells, the cells can separate and the embryo will still be able to develop from any of the cells. Does that make sense? That is, these cells come together to give the one embryo. Why? So because of that, they don't separate. So once there is cleavage, When there is separation of these cells, then the embryo cannot be formed again. But in this case of mosaic, the non-mosaic or indeterminate cleavage, the separated cells can still individually give rise to different embryo. All right, so that's that about cleavage. So there are just two of those type of cleavage. Then the development, okay? Development, based on development, organisms are characterized into organisms either have direct development or indirect development. When an organism has direct development, it means both the larval stage and adult stage, larval stage equal to adult stage in resemblance. So the larva looks exactly like the adult. But in indirect development, there is metamorphosis, a stage known as metamorphosis. Okay? And what is metamorphosis? A gradual change in form and structure of, and of course, physiology of an organism over time until it gets to the adult stage. It starts from the larva stage and it undergoes series of both physical and physiological stage changes until before it becomes the adult. So larval stage here do not look exactly like the adult. So and because of that, it gives them an advantage though. The advantage it gives them is that they are able to have they have vagaries of resources that they can make use of at different stages of their life. So because of that, they don't really have problem of depletion of resources. Unlike these guys, whose larval stages and adult stages are very similar. So because of that, there is possibility of high competition and depletion of resources. Unlike these guys, who the different forms that they take at different stages of their life have different things on which they feed on. Does that make sense? All right, so that's an advantage for them. Just like I wanted to mention an advantage of motility, yes. So for example, organisms can either be, for example, when we're classifying as based on symmetry, yes, I remember now, symmetry. You know, we said that some organisms are not symmetrical at all, while others have radial symmetry. And we mentioned that those ones that have radial symmetry, many of them are sessile. So, and that's, sessile means not moving, right? So, for those that didn't watch the video. So that radial symmetry gives them an advantage of being able to access different resources in a place. But cephalization, I didn't mention that, so cephalization is part of the basis of classification, or features of classification, basically. So cephalization came as a result of bilateral symmetry. So so we call them bilateralian. So bilateralians have the word cephal or cephale is the Greek, I think, maybe Greek or Latin, I think it's Greek, for head. Is that clear? So what happened is that those bilateralians, yes, have head. They began to have head. Why? Because the sense organs became associated or became close to one another in one portion of the body, so that that portion will be the portion that can get food or move towards food. So the necessity for mobility came. Does that make sense? The organism had to be motile, or motility rather. The organism had to be motile because only one portion of the body now can now is adapted for sensing food, for location of food. So because of that, there is cephalization. So all the senses are aggregated in one portion of the body that is now called the head. So the same way that became an advantage, and of course radial symmetry is also an advantage for those guys that they don't need to move, before they can access, even though some actually move, as we have in echinoderms. The same way here, direct, indirect development is also an advantage for the organisms. Examples are arthropods, insects primarily. But almost all, in fact, all arthropods actually have indirect development. No, almost all, because exceptions do exist. Exactly. So from crustaceans to arthropods, arachnids, and all of that, they are all arthropods. Then they all undergo indirect development. While we see direct development in mammals, All right. Then all of these guys from here downward, we have indirect development. Even with mollusks, we have indirect development, except in a few. There is direct development in a few of them. There is direct development. So then arthropods, indirect development. Echinoderms, indirect development also. But chordates, mostly direct development. Okay. Then the type of egg. So the type of egg that organisms lay, we also use it to classify them. So I will classify them into three. We have organisms that have microlecithal eggs. Let me confirm that this thing is still recording. Okay. We have organisms that have microlecithal egg. The word micro, what does it mean? Small. Lecithal, the word lecithos. Lecithos is the Greek for yolk. Lecithos. Okay? So microlecithal. You already know what that means, yeah? Mesolecithal, and macrolecithal. So the microlecithal will have small, right, yolk compared to the egg size. And of course, because of this, the size of the organism that will develop is usually small. Okay? Mesolecithal, that is, there is equanimity, and there is like equal yolk to egg size. Exactly. That is, the yolk is not small, it's moderate. Okay? Yolk is moderate yolk size. Macrolecithal, large, yeah? Large yolk size, okay? And in fact, these ones are not divided into like 3 or 4 or 5 based on the position of that large yolk. So we have some that are, that are positioned on one vegetal end of the egg. We call them the telolecithal. So that is, the yolk is shifted to one vegetal end of the egg. Then we have the myolecithal. The myolecithal, that is, there is the yolk fills the entire egg perfectly or moderately, but there's a membranous disc on one end. Then we have the central lecitor. This is one in which the central, meo means incorrect measure. Exactly. Telo. The word telo means end. Exactly. Central, center. So that is the egg is the yolk is exactly at the center. The large yolk is exactly at the center. We also have about 4 or 5 of them. We have the telolecitor on one end, in the middle, then filling the entire thing. Okay, so that's basically it. All right. So for the microlecital, we have the mollusks. They are under this. They have microlecital eggs. Then mesolecital eggs, we have some fish, so some chordates. And microlecital, we also have amphibians. Amphibians. Exactly. Then macrolecital, we have chordates. Birds are under macrolecital. Okay. So that's that. The next one is body temperature. The body temperature is organisms are divided into two based on the body temperature. We have the homo, homo, um, homeotherms. Let me use the word endotherms and ectotherms. But I'll still have to mention it. So exotherms. So endotherms are the ones that do not rely on their external environment for their body temperature, and we also call them homeotherms or homokilothermic. Now, what this means is that they they regulate their internal temperature by themselves, so it doesn't depend on the external temperature. Okay, so for example, mammals are endotherms. But exotherms are the ones that depend on their external environment for the regulation of their body temperature. Okay? So an example is the fish. We say they are poikilothermics. Poikilotherms. Poikilothermic. So that is, they are cold-blooded. Yeah, that's another word. These ones are called warm-blooded. Warm-blooded. While these guys are cold-blooded. Cold-blooded because they depend on the external environment for the regulation of their internal temperature. So the external environment is cold, so their blood will be cold. Okay? All right. And for us, our blood is usually warm. Okay? So that's that for body temperature. Another is digestion and digestive system. Okay? Digestion. Now, we either digest things internally or externally. That is, animals rather, either digest things internally or externally. We call it internal digestion, as we have in us and many mammals, and we have external digestion. External, um, external digestion. In this internal digestion, we mean, internal digestion. This one, in this external digestion, we mean extracellular digestion. In this case, the organism will secrete something The organism will secrete um, an enzyme that can help in the digestion of that thing. So after it digests the thing, then it absorbs the nutrients. Is that clear? Using its body. Does that make sense? Okay. So that's extracellular digestion. And an example of organism that does that will be netophores, coelentrates and netophores. They digest things extracellularly. Coelentrates and netophores. Some mollusks, coelentrates and netophores. Some mollusks, they are they engage in extracellular digestion or external digestion. All right. The next is digestive system. Okay. Now the type of digestive system we have complete and incomplete digestive system. In the complete digestive system, example of what we have for the complete digestive system is we have a mouth and an anus. So you will know why we split organisms into what? Protostomes and deuterostomes. Don't forget it. Proto, we said early, stoma, mouth, right? So it means that the first opening, the first archenteron, or the first blastopore forms the mouth for protostomes, while the second one that appears forms the anus. But for deuterostomes, which I already told you also, protostomes are these guys. These organisms that are triploblastic are the ones that we divide into this. Is that clear? So this is a protostome, 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 protostome. Then this guy is a deuterostome. From there downward, deuterostomes. So, so I was talking about complete GIT. Okay. So, but you see, these guys, the platyhelminths are protostomes, but they have incomplete gut. They have only that first opening, only the archenteron. It is only the archenteron that they have as their embryonic opening. They don't have another opening, which is supposed to be like this. They have what we call blind gut. So this one opening serves for both entry of food and ingestion of waste. So platyhelminths, they are the only ones here that have blind gut or incomplete GIT. Is that clear? All right. Then let's talk about respiration and respiratory system there. Now, respiration, animals respire through various structures. Is that clear? So, based on respiration, we classify them as the different types of respiration. Okay, first, let's use the aerobic and the anaerobic. Okay, aero, that is air, right? Or oxygen. So it means that these organisms use oxygen, yeah? While the anaerobic, they don't require oxygen for respiration. They don't use oxygen for respiration. Example of anaerobe is this platyhelminth. The rest of them are aerobes. I hope that makes sense. That means chiefly or primarily they use oxygen, because it doesn't mean that we don't engage in anaerobic respiration at all. Only that most of the time, or chiefly, our major energy source is via aerobic respiration. While for platyhelminths, they engage majorly in anaerobic respiration. Right. So, and then reproductive systems or reproductive structures. So these ones, they have um, um, you know, a water current system. A water current system. It brings in water via the ostia and takes it out of the um, osculum. They have only one opening, exactly. Then um, the ones that necessarily breathe in oxygen and breathe out carbon dioxide. Um, we have them to be the platyhelminths. Platyhelminths, there's something I want to remember. The so so these ones by simple diffusion, exactly. By simple diffusion, they respire. This guy also respire by simple diffusion. Exactly, using the body surface. Because for these organisms here, many of them have a lower um, surface area to volume ratio. Their volume is smaller than their surface area. So, but the um, from the annelids, the arthropods rather, from the arthropods, we begin to see different structures. Mostly for the insects, we see for the insects, and this is annelid, sorry. Annelid also respire through body surface, F1, body surface. I wanted to talk about arthropods. No, from these guys first, mollusks. Mollusks use ctenidium or primitive gill. Is that clear? It's a primitive type of gill that they use for respiration, especially the ones that are that are aquatic. While the lands, they will use what looks like their ctenidium has been modified into what looks like lungs. Is it clear? So ctenidium for most mollusks. And of course, there is also a water current exchange system for some other mollusks like octopuses, and bivalves. Okay. Then this guy, arthropods, those guys, they use a tracheal system. So these ones, they use what is called a branchial system. Branchial system is a system of gills. Is that clear? While the arthropods use a tracheal system, that is, they use oxygen permeates their body through holes in their body called spiracles. Then these spiracles make it into the body through the trachea. Is that clear? So we call it a tracheal system. And this trachea leads to tracheoles, and these tracheoles have direct connection with the body fluid known as the hemocoel. Hemocoel. I didn't mention under coelom, there are three types of coelomates. We have the schizocoelom organisms, and we have the enterocoelom organisms. We are under the enterocoelom, the chordates. Then we have the hemocoelomates, these coelomates. So these guys, arthropods, insects primarily, are under the hemocoelomates because their own mesoderm, their mesoderm separates their coelomates exactly, but their mesoderm actually arises from the lining of the internal gut, the endoderm, the the schizocilomate. But the hemocilomate, the space is now filled with the coelom is reduced. The coelom of hemocilomate is reduced because hemocoels, the body fluid, which we call hemocoel, fills the space up. So the coelom is reduced. And we call them hemocoelomates. Then the enterocoelomates is where we, chordates, exist, where we are. So, and the schizocoelomates are just the organism coelomates that their mesoderm splits after the enteron the enteron invaginates inward. Invaginates. Yeah. So the mesoderm is formed from when those cells split apart. Okay, so the mesoderm splits into two. So those are schizo. The word schizo actually means split. Enteron, to enter. Does that make sense? Hemo, related to the blood. Is that clear? Enteron, that is invagination, something that enters inwards. Is that clear? Schizo split. Okay. The video is already long, so please make sure that you subscribe to the channel. And please, if anything at all, do not miss the question and answer part of this video, of this series. So that's for respiration. Let's talk about, and the respiratory system. Okay, I'm still on that. These guys, echinoderms, they use what we call tube feet. It's also a water tube system, water current system, tube feet. So it takes in oxygen and, takes in water, and then they filter the oxygen out of from the water. Then the chordates use different things. Now, depending on the chordates, yes, depending on the class it belongs, we use different structures. Okay, so if you are in the water, like in Pisces, you use gills. Amphibians will use external gills when they are in their primitive, their infant, their embryonic stage, not embryonic, and their larval stage. That's the word. I'm sorry, larval stage. So amphibians will use external gills in their larval stage, and they will use internal gill for some adults and cutaneous respiration, or gill or skin, through their skin. We call it cutaneous respiration. And some lungs. In reptiles, primarily lungs. Aves, birds, primarily lungs. Then mammals, lungs. All right. So that's that for respiration. Then for excretion, organisms are split into three based on their mode of excretion, right? So we have the amonotelic organism, amonotelism, urecotelism, and ureotelism. So we are ureotelic, chordates. No, not chordates, like mammals, I'm sorry. Mammals, because not all chordates are ureotelic. Basically, these words are from the word ammonia, right? Ammonotelic, okay? So it means that they are major nitrogenous waste. You already know what excretion is, right? Removal of waste product of metabolism. So ammonia is their nitrogenous waste. They are major. Now, this does not mean that it's only ammonia they excrete, but it is their major Their major nitrogenous waste. Uricotelic. Uric acid is what they majorly excrete. This guy, ammonia as their major nitrogenous waste. These ones, uric acid as their major nitrogenous waste. And this one is urea as our major nitrogenous waste. Uric acid is excreted by reptiles, for example. Urea by mammals and birds. Urea. Uric acid by reptiles. Ammonia by Pisces and amphibians, yes, and some mollusks, ammonia. From here, ammonia, downward like this, ammonia. While some arthropods, yes, some arthropods, depending on their environment, you know, there are some in water and there are some outside. So depending on their environment, they either excrete uric acid or ammonia. Okay, so that's that. That's how we classify them. But as we go down like that till we get to this place, they are majorly ammonotelic. They're majorly ammonotelic. Okay. So that's that. Then there are excretory structures. Excretory structures. These guys also excrete by simple diffusion up to this point, up to netophores, simple diffusion. This guy excrete by diffusion and now platyhelminths. So you must know this. Platyhelminth is a very common question. Platyhelminths, they use flame cells. See, if you usually forget, it's very simple. You remember platy means flatworms, right? Flat. So flatworms were excreted by flame cells, FF. Is that clear? Then we have nematodes. Nematodes, those are aschelminths. They excrete using H cells, right? Or tube cells, tubular cells. Tubular cells. Then we have annelids. Annelids excrete by proto or early or primitive nephridium. Now, this flame cell is actually like a nephridia too, but we call them flame cells because of how their motion, I will link the video in the description where I talked about excretion. I've initially talked about excretion, and I used comparative anatomy of this by evolutionary advancements. Annelids. Then mollusks. Mollusks, they use different things, but majorly nephridium. Majorly nephridium. Nephridium. Then the arthropods, they use the arthropods, depending on the arthropod. Some use green glands, such as we will find in some crustaceans, spider rather, green glands, spider, no, longbook, spider, crustacean, green gland, longbook, spider, insects, they use Malpighian tubules. They're just tubes. When you hear these tubules, it means tiny tubes, right? Insects under the arthropods. Okay? Then chordates, like I said, we use these guys. They use kidney. Chordates use kidney. Yes, they all have kidneys. Yes. So that's that for excretion. Then circulatory system. We have two types, the open and the closed circulatory system. The closed circulatory system is seen only in chordates. The rest of them have open circulatory system. That is, there's direct connection between the respiratory system. That is the blood fluid has direct connection with the oxygen and other organs. Exactly. But in closed system, there is a tube such as artery, vein, and capillaries via which the blood will flow. So someone will now say, well, sometimes when I cut myself, I don't see the tubes. The tubes are inserted into your epithelial, your skin, inside your epithelial tissues. So, if you cut through, you won't know you've cut them. They're so tiny. All right. So that will be it for this video. Do well to see all the videos. I'm going to link in the description. We're going to go immediately to the classification and the characteristics. God bless you as you subscribe.